You know how some bacterial infections are easy to treat, while others are super stubborn?
Well, it often comes down to one fundamental difference in how they're built.
You see, bacteria are split into two major groups, and it's all about their cell walls.
So gram-positive bacteria, they have this thick, mesh-like armor called peptidoglycan.
But gram-negative bacteria have a much thinner, more flexible layer of that same stuff. Gram-positive walls also have these things called teichoic acids woven in for extra support. You won't find those in gram-negatives, though. They have a totally different trick up their sleeve.
That trick, it's an entire extra outer membrane. Think of it like a second protective skin.
And this outer layer contains lipopolysaccharide, or LPS. It's a seriously potent endotoxin. All of these structural differences are exactly why they stain differently in the lab.
Because this structure, it dictates exactly how they need to fight them.
See? That thick wall on gram-positives is surprisingly vulnerable to antibiotics like penicillin. But that extra membrane on gram-negatives, it basically acts like a shield, blocking many drugs. Common gram-positive baddies include things like staph infections and strep throat. And on the gram-negative side, you've got E. coli and salmonella. That LPS toxin is no joke. So yeah, everything from the lab result to the right medicine, it all depends on that wall. All right, let's put it all together. Here are the key differences side by side.